And good afternoon, good morning, molecular genetics friends. So let's see, we have a guest lecturer today talking about her introns. This is my friend Quinkle. Quinkle, say hello. She's not sure what to make of online teaching either. We're going to be talking about chapter 4 in the book. And I don't know if you have the book or not, but it is on introns. And the reason we want to get into this is our next sort of discussion topic is going to be on genomes and genome evolution. And introns are a real critical part to this in terms of complexity, the ability of taking a gene that has introns, we can actually do differential splicing, and that's a fairly big deal. And as we're going to see, when we start looking at more complicated eukaryotes, the more complicated you are, the more likely you are to have multiple introns, and that does allow for differential splicing. I'm going to do something a little bit different today in that I'm going to be running through some PowerPoint slides simply because there's some graphs that are pretty nice to look at and some diagrams that are from the book, and it's a little bit easier to do than me writing on the board. So there you go. And I'm going to kill the lights a little bit to bring up the contrast. And so hopefully you got a little bit of insight into some DNA sequence analysis by looking at the PCR primers. And let's see, we're coming up. Yeah, wait for it. Wait for it. Here it comes. We're looking at the interrupted gene. I wish I had a way I could write on the board at the same time. There probably is some sort of doomed virtual way I could do it, but
it's the B key.
That's all right. All right. So we know in eukaryotes that the RNA, the mature RNA, is not a direct copy of the DNA, that there are in fact these regions that are interrupting the open reading frame. And these interrupting sequences were identified when people began to compare the RNA molecule of a gene with the DNA molecule that was the transcription template. And what you begin to see is these little squiggles, actually using electron microscopy, they were able to see that the RNA was like a line, and then the DNA underneath it would have these loops. And these loops, of course, were the DNA sequences that were not present in the RNA and didn't hybridize. And so what we have then is this term that was known as intervening sequence. And so it became known as an intron. And then the other part that was not intervening, that was the mature RNA, is known as the exon. So when we think about the structural gene, that is the portion of the genome that is being transcribed by RNA polymerase. In prokaryotes, the structural gene is the messenger RNA, or it is the DNA form of the messenger RNA. But in eukaryotes, it isn't necessarily that way. Now, there are some genes that don't have introns, in which case the structural gene is the same as the RNA. But most genes are going to have at least one or more introns. And let's go ahead and see what we can make from this. So here is a cartoon of a piece of genome, and we've got exon 1, intron, exon 2, intron, exon 3. So the RNA synthesis is going to generate a very long RNA, what we can think of as the pre-messenger RNA. And then the messenger RNA is going to be processed. We've mentioned this before. It's going to be the 5' cap, the 3' poly A tail, and splicing. And these things are kind of occurring somewhat simultaneously. As the RNA after it's been transcribed, it can now be processed. So we then have the fully processed messenger RNA, and this messenger RNA then can be translated to generate the protein. So a couple of things. We still have untranslated region at the 5' end and 3' end. We still have the AUG, the initiating methionine, embedded in that RNA. So it's not the splicing doesn't generate a ready-to-go translated sequence you can just zip through. You still have the 5' untranslated region where the ribosome is going to interact as we get into translation. Now, another aspect of this is why we've talked a little bit about cDNA, it's complementary DNA, where you take the RNA and turn it into a DNA form so you can clone it. When we're trying to take a genomic region and express it in bacteria, the bacteria, the prokaryotic system, can't splice things out. So in order to have a gene expressed in bacteria, it has to be fully processed. All right, so a couple of points. I'll post these PowerPoints also on the Moodle. But the introns are removed by the process of RNA splicing. Mutations in exons are going to affect the protein sequence. Mutations in introns, as we mentioned before, a common single nucleotide mutation is a mutation that affects the splice site. There are these invariant nucleotides at either end of the exon. And if they get monkeyed around with, you change them, that exon or that intron is not spliced out. It's going to be just contiguous. And as we'll see here, introns will almost always change the reading frame and introduce a number of stop codons. So mutations in introns can affect the RNA processing, and that is how they can actually function as a mutation. So the primary rule then is we can have a whole series of exons and do differential splicing in which we can drop different parts out, but no matter what, the intron or the exon order has to be preserved. That is, you don't have the ability in this case of having exon B ahead of exon A. It's always going to be in the same order, but we can do differential splicing in which we could have multiple RNAs, A and C, or B and C, or AB, or any other combination you think of. So as long as you're maintaining the order, the splicing can actually be a whole range of things. So the exons remain in the same order in the messenger RNA as they are in the DNA, but the distances, because you're removing things, aren't going to be the same. Now, the interesting thing we should start thinking about genomes is that very often within a gene, looking at it in a variety of different species, we'll have the organization being preserved. So the exon-intron organization of genes is often very well preserved. And really the way you have to determine what is an exon and what's an intron is by comparing the RNA with the genomic DNA. And as we're going to see as we start looking at more complicated eukaryotes, that the RNA that a gene produces, there can be multiple RNAs, multiple species, because of differential splicing. All right, so one of the ways there, like the initial ways we mentioned, is doing a hybridization experiment where you take RNA and you hybridize it with DNA. And when you do this, the intron isn't going to be hybridized to the RNA because that sequence has been pulled out and you'll have this loop. Now, an experiment you can do is something known as an RNase protection assay. And let's see, how would that work for this? What you would do is you would take the genomic DNA sequence and transcribe it. You can do this in vitro. You would then take the cDNA that you're working with, hybridize those two molecules together, and you'll get this loop structure, and then use RNase, which will break and degrade single-stranded RNA, but not double-stranded RNA. And then you can run the products out in a gel, and you can look to see what you got. You can begin to identify these different fragments, these different portions. The other way this is carried out is actually by sequencing. So you sequence the cDNA, and then you compare that to the genomic DNA sequence. So, as we mentioned, the positions of the introns is generally conserved, looking at homologous genes. The length can be tremendously different. Introns are not going to typically be functioning as any kind of gene or information. As we said, the reading frame is going to be pretty much generally expected to be interrupted. That is, there's going to be stop codons in the frame that would be going along when you get into an intron. And so when you're looking at DNA sequence and you have a genome or a genomic DNA fragment and you run a translation program through it, you'll see good open reading frame, and then you'll see a spot where there's a stop, and it'll absolutely just be stop after stop after stop. So introns, these intervening sequences, they are always filled with stop codons. And what we're going to do is actually link together these different pieces and generate a contiguous open reading frame. So another thing that is really cool that we actually mentioned in the evolution discussion a bit, an evolution lecture for Jerome, hey, name call out, you're in evolution. Not surprisingly, exon sequences are generally quite conserved, but introns are not under any kind of selective pressure other than the invariant sequences required for splicing. They're not under selective pressure, so introns between species can be phenomenally divergent. And this actually can be used as this notion of an evolutionary clock, in which you can look at the length of time that two species have separated by looking at the number of nucleotide differences in the introns compared to the exons. So because the exons are the actual coding region, they are going to be evolutionarily constrained. But the introns, man, they can go crazy. And so we have all kinds of differences in introns. So when we look at a gene sequence, we can have this dot plot that you guys actually were looking at with our alignment assignment. And you can do that either with amino acids, you can also do it with nucleotides. And when we look here, we see that looking at two homologous genes that are closely related within the mouse in this case, they've got very conserved regions in the different exons, but in the introns we have significant sequence divergence. And this makes perfectly good sense here. All right, so we're going to begin thinking about genomes on a bigger scale. And when we start thinking about this, one of the things we compare is we have these model systems that we kind of look at in terms of complexity, looking at bacteria, and then in this top panel, this is Saccharomyces cerevisiae, that is the standard bread yeast. We have our friend Drosophila melanogaster, the fruit fly, representing all of the invertebrates. And then we've got mammals, you and I, an elephant, whatever. And the interesting thing to look at here is we know in prokaryotes there are no introns. In single-cell eukaryotes like yeast, which are just a notch above prokaryotes, most of the genes are in fact still uninterrupted. There are a very small, 8% or so of the genes that have a single intron. If you look at the x-axis, this is the number of exons. So the number of introns is the number of exons minus 1, maybe? Anyway, so having 2 exons means you got 1 intron. Now, when we start looking at a slightly more complicated organism, Drosophila, the number of single uninterrupted genes drops dramatically, from 96% down to 17%. And we've got a bunch of genes that have a single intron. And then when you go to mammals, you and I, the number of genes that are not containing introns at all is down to 6%. And really, 2 is pretty small as well. Start seeing a lot more introns here, 5 being probably the average. And then if we look out here, there are some that have a tremendous number of introns. Now, yeast really don't have anything that's that complicated, but flies have a few genes that have got more than 10 introns, but we've actually got a number of them that are really quite complicated. What this means is that you've got a lot of genomic real estate, of genomic DNA that is present, and then it gets spliced out phenomenally complicatedly to produce a much smaller RNA. So the take-home point here is the more complicated you are, the more introns your individual genes will have. And this then can be seen as well by looking at the gene size, in that because of the introns, the gene size can change. And when we look at the exon length, if you're going to end up having lots of introns, your exons end up being a good bit smaller. So you chop things up. Because, if we think about this, yeast has essentially the same proteins as the mammal, when at least all the individual proteins are going to be very similar. Actin's going to be the same. The different Krebs cycle enzymes, whatever, those amino acid sequences are the same. Because they don't have introns, those genes are going to be bigger. But when you start looking at the gene sequence of or the exons in a organism that's got splicing, we end up with a much smaller kind of exon size here. So with yeast, the exon is the entire gene, and with human, the exon is a small piece. All right, so introns generally are fairly short, but as we said, the intron is not subject to evolutionary pressure, and so they actually can be quite large. And in general, the more complicated the organism is, the longer the intron ends up being. So there are some genes, and we're actually going to be looking probably next week. I got to actually do a little bit of thinking about the DNA structure before I give it to you. The gulolactonone oxidase gene that is involved in vitamin C production. I'm kind of thinking about figuring out my PCR protocol for this and looking at it in a couple of different organisms. And we'll see that there are differences in the intron length within different organisms, and actually look to see how we can use that in a cloning strategy or in a sequencing strategy. So yeast genes are short, but genes in flies and mammals are longer, mainly because we start to have a greater number of intervening sequences. I mean, this makes pretty good sense here. All right. Now, one of the things that happens is that with intron-exon splicing, we can have a single gene produce multiple gene products. Now, there are a couple of different ways to think about this. We can have alternative splicing that can actually add or subtract different amino acid sequences. Sometimes like in the middle of a protein. One example might be you could have a different enzymatic domain that might be present or absent. Another standard thing that a lot of eukaryotic genes have is alternative start sites. That is, the AUG can be different in that we've got an RNA that has a 5' untranslated that we're not looking at, and we've got the AUG start, but then we can have a splice that will remove this part of the RNA, and now the initiating methionine is going to be further downstream. So methionine, of course, is coded by the triplet AUG, and that is typically the first amino acid of the protein. There are going to be sequences that are going to line up and say, all right, this is the appropriate RNA start site for translation. This is the appropriate RNA sequence for start. But you can have multiple AUGs, inside a gene, and what you have to do more or less experimentally is determine what is the start site of the amino acid system here, the protein, and that can be typically done by looking at the size of the protein product that is predicted versus the size that you see. And so what we got here, though, is the possibility of having a protein that starts with an AUG here or a protein that starts with an AUG here. And a standard kind of rearrangement that can occur is one that may require sort of a throwback or a callback to Molecular Cell. We talked about the fact that there are sequences, generally hydrophobic sequences, at the amino front end of a protein that are part of the Golgi directing kind of sequence. It's called the signal sequence. And that's what directs a protein to be translated into the ER and then make its way into the Golgi. So this is the signal sequence that is the protein localization sequence that's going to direct a protein into a very specific spot in the cell. There are proteins that can be, where their genes are differentially spliced, and that you can remove a signal sequence. And so we might have a protein that is going to be in the cytoplasm, or a protein with a signal sequence that's going to end up in the Golgi and maybe end up being secreted or stuck in the membrane. Another thing you can think about is you can have a protein with a nuclear localization signal. If the nuclear localization signal is present in the protein, then that protein is going to get directed to the nucleus. If that exon has been removed, we can have the protein with all its function now without the nuclear localization signal in the cytoplasm. So differential splicing can produce proteins that are almost completely identical but have different localizations. So that's one way we can think about the diversity that introns can generate. Now, another thing we can do is have really completely different proteins when we've got a change in the exon-intron splicing that results in a different reading frame. That's a fairly rare thing. You have what are these overlapping genes. More generally, we can have differential splicing that's going to result in portions of the protein being similar and then portions that are added or subtracted. So alternative splicing is a really critical point to think about, in that we can generate multiple protein species from a single gene. And just as an example here, we have exon 1, exon 2, exon 3. So we splice things out and we end up with a long protein that has all 3 exons, or a shortened protein that has only exon 1 and exon 3. Quinky, what are you seeing out the door? You're all right, girl. Yeah, come over here. All right. All right. So, one of the things that we can begin thinking about that actually does seem like it has some legitimacy is that the exons can be equated with protein domains, with these functional domains. So you have a kinase region, a region that's going to act as a kinase. You can have these different spots for directing protein localization. You could have membrane-bound versus non-membrane-bound forms of a protein. You could have different kinds of anchoring or binding proteins. A whole range of different kinds of ideas come about here by being able to take a protein and change some of the different kinds of functional regions in it. So not all the time, but very often exons will have a functional encoded region in them. And that allows you to actually sort of do mix and match of different kinds of functions within the same gene. It's also an evolutionary tool in that we can begin to see exons that are present in multiple genes through recombination and things. So when we look at things like the immunoglobulin gene, and we'll get into the development of the immune system in a few weeks, the immunoglobulin gene is developed through sort of a mix and match of dropping different pieces out where you've got a range of different functional parts, these protein domains, and we can do differential splicing to generate a single protein where you've got multiple different kinds of exons to choose from. And it's a way of generating a single protein that's got a significant amount of variety to it, but it still maintains these different functions. We have a variable region, a constant region that's always there, the hinge region, constant regions, and then the constant region, and they can be different exons. And so you can build a protein up with some variety in it this way. Immunoglobulin genes are kind of weird, though. They undergo some very significant splicing and rearrangements, but we'll get to that. All right. One of the things that we've been looking at with the DNA sequence and amino acid sequence homology is this idea that's known as a gene family. So within a genome, when you begin looking at the genes within a genome, you can begin to identify genes that have within an organism a lot of similarity. Like for us, we have a variety of globin genes, and they're expressed at different times during fetal development, and then there's the adult form of globin. We've got multiple actin genes. We've got multiple myosin genes. There's muscle myosin and non-muscle myosin. And they're going to often have real similar sequence structures, and they even have exon-intron splicing structures that are in common. And it suggests that these different individual genes, they're members of a gene family, arise from a duplication event, which is actually probably the way these things actually do arise. So the exon-intron structure is associated then with these various kinds of functions. And we can see that the domain is divided here, but for the most part, we've got exon-intron structures that are being retained to generate two different proteins of very similar kinds of amino acid sequence. Is that the last one? Maybe the last slide. All right, so the take-home point then is that the more complicated the genome is, and the more complicated the organism is, the greater the exon-intron structure is. And this is going to contribute, as we'll start seeing, a lot to genome size. And that's all I have to say. So we're going to start looking at some genomes and thinking about, now that you have some ideas about doing PCR primers, developing some PCR primers to see what kind of amplifications we can do of different genes, and actually do a little bit of in silico, as it's called, on the computer PCR, and see how that works. All right, that's all for today. I will check in with you guys on Thursday, and we'll see you.